Now let us begin with the anti-protozoal drugs. First, we know that let us see the screen. Now, what are the anti-protozoal drugs and give its classification? This is what the sort of question you will be asked in the examination. So, in that, the anti-protozoal drugs or the anti-protozoal drugs definition would be, it's as simple as this: the drugs which kills or inhibit the growth of organisms known as protozoans. So the drugs which will either kill or inhibit the growth of protozoa, that we call it as an anti-protozoal drug. In the last lecture we had also discussed about the protozoa. What are the protozoa? These are the single cell one, the single cell eukaryotic that we had discussed. Then they are either motile or non-motile. This also we have discussed. And these are nothing but the parasites. They are either living or non-living. That also we had discussed. And in the last lecture we had also come across this one, that is they causes the amoebiasis, leishmaniasis, even the trypanosomiasis, and caused due to what? The consumption of contaminated water and food, and it spread through some vector, especially the insects or mice. So this is what we had discussed in the last lecture. Now today we are more focusing on the anti-protozoal drugs. Now, classification of the anti-protozoal drugs. So first we will begin with the classification of anti-protozoal drug. See this one, the anti-protozoal drugs classification. The classification we can make into two things, that is, one is according to the site of action where these anti-protozoal drugs are going to act. That is what? The site of action. And of course the second part is the according to the chemical structure, that is what the chemical moiety which is present in it. Now coming to first one, that is the according to the site of action. Now the first, it would be the luminal amoebocytes. Now first one is a luminal amoebocytes. That is what? These are used into the intestine. Understand? So that is the intestinal amoebiosis. You know that intestinal amoebiosis that we had discussed in the last lecture. So for intestinal amoebiosis, the drugs which are used, that we are called as a luminal amoebocidal drugs. So these drugs could be dioxanides, then your this diodrohydroxyquine, then iodochlorohydroxyquine, even the iodoquinol, then these are pentamidine isethionates are there, even the atovaquones are there, and the dimercaprol. So these are the various drugs that we are going to use it as a luminal amoebicide, that is to cure the intestinal amoebiosis. Understand? So next could be what? The next is the that is the tissue amoebicides. Now in case of a tissue amoebicide, they are used in the not in the intestine, that is apart from intestine, in other tissues. So wherever there is the protozoal infection other than in the lumen, so that is the tissue amoebicide. The best example would be your malaria. So the drugs which can be used are these one, that is the chloroquines are there, even the imatines are there, and the dihydroimatines are there. So these are the drugs which we use it as a tissue amoebocidal drugs or tissue anti-protozoal drug. And apart from this, there are something which is called as a mixed one. Mixed amoebicide means what? They both are used either in the lumen, or it can also be used in the different types of tissues as well. So the best drugs in this particular category are the metranidazole, tinidazole, and the paramomycin. So metranidazole and tinidazole are very important drugs. Now coming to the according to the chemical classification, if you consider the chemical classification, then according to chemical structure, so one could be the first could be the alkaloid. So example for alkaloid are imatidine or imatidine bisulfate, or that is the bismuth iodide. Okay, so these are the alkaloids. Then the next one category is the halogenated hydroxyquinolones, chloroquines. Understand? So this is the another class. Then the third important class, which is very important for exam perspective as well, so that is the nitroimidazole, 5-nitroimidazole derivative. So nitroimidazole derivative, that is metranidazole, tinidazole, ornidazole. So these are the drugs which we are supposed to study. And some organic arsenicals are there. So these are the various types of the anti-protozoal drugs. Now once we understand the anti-protozoal drug, then we can move to the next part, that is the we'll discuss about the protozoa, I mean the metranidazole. Understand? So the question will go like this, that is they give structure, IUPAC, MOA, that is mechanism of action and synthesis of metranidazole. Please understand, I'll repeat once again, give structure, IUPAC, mechanism of action and synthesis of metranidazole. Understand? So now see the structure of how the structure of metranidazole will look like. Please see. Now this one is the structure for metranidazole. Now in this metranidazole, if you can see, the first number is given to nitrogen, second number understands, then second, third, fourth and fifth. So at the fifth position you are having nitro group. So the nomenclature, the basic ring, what the basic ring could be, the basic ring is 5-nitroimidazole. What is imidazole? The 5-membered ring with 2 nitrogen, we call it as imidazole. Understand? And at the fifth position we are having nitro, so that this would be the 5-nitroimidazole ring. Apart from that, at the first position, what is there? At the first position, hydroxyethyl group is there, right? And at the second position we are having the methyl group, so that we can say 1-hydroxyethyl, 2-methyl, 5-nitroimidazole. So this could be what? This is nothing but your IUPAC name for metranidazole. Understand? So that is the me refers to what? Now in this one there is a me is nothing but the methyl group which is present at the second position, right? And that of the imidazole, that is the dazole comes from imidazole. Understand? And ni is the nitro. Understand? So nidazole is 5-nitroimidazole. Understand? And Me refers to what? Me refers to your methyl group which is present at the second position. So you should be in a position that while that particular name utters, you should be in a position to draw the structure. Metranidazole, nidazole is nothing but the imidazole ring with the nitrogen. Ni is nitro, so nitroimidazole ring, so that you will draw. Me is methyl you will draw. Understand? So likewise you should be able to recall the structure for metranidazole. Now this metranidazole is actually acting as a prodrug. What is prodrug? The prodrug is actually the drug with the something pro-moiety, and once it enter into the system, it will undergo chemical cleavage or enzymatic cleavage or some certain reactions to release the active constituent. So similarly in this also, metranidazole, this particular metranidazole, what is happening, you know, this metranidazole, the prodrug, now what will happen, this will undergo the reduction reaction. That is what is happening, you know, that is the nitro group is converted into the hydroxylamine group. Please understand, this metranidazole will undergo the reduction reaction by the simple electron transport chain. Understand? And because of this, the nitro compounds or the nitro group will be converted into the hydroxylamine group. Now this hydroxylamine will get covalently bound to the DNA, and as it is bound to the DNA of the microorganism, that is the lethal to that microorganism, that is the lethal to that your protozoa. Now similarly, when this nitro group is getting converted into hydroxylamine, during this particular process there is the release of the reactive oxygen. There is the release of the reactive oxygen, and what this reactive oxygen will do, it causes the destructive effect on the cell component, that is the DNA proteins and even the membrane protein. What it does, it destroys it. So this is what the mechanism of action for metranidazole. Now after this one, the important part is the synthesis part, or the synthesis of this particular compound. Okay, now let us discuss the synthesis. This is very important one. Now metranidazole synthesis will begin with the glyoxal. What is glyoxal? Two aldehyde groups which are connected to each other, CHO CHO. This is glyoxal. And what it is used, what again the second thing what it is going to use, that is the ammonia part and that of the CH3CHO. What is that? Acetaldehyde. So that during the synthesis of metranidazole, one acetaldehyde, two ammonia molecule and the glyoxal, these three things are used. When these three things are used, you will get the cyclized compound. This is the cyclized compound. This cyclized compound is nothing but what? This particular cyclized compound, see this one, this is the cyclized compound. This is 2-methyl-imidazole. That is the cyclization took place. Understand? So see, these two nitrogen come from where? These are the two nitrogens. Understand? 3 water molecule lost. Understand? And this methyl comes from the acetaldehyde, and you will get the 2-methyl-imidazole. Now once you get this 2-methyl-imidazole, this 2-methyl-imidazole will undergo nitration reaction. Nitration reaction is what? Reaction with the nitric acid and sulfuric acid. Now once it undergoes the reaction with nitric acid and that of the sulfuric acid, you will get the second substitution, very important substitution. In the first reaction you get what? 2-methyl substitution. In the second reaction you will get 5-nitro substitution. So that means the compound what you are getting right now is the 2-methyl-5-nitro-imidazole. Now this 2-methyl-5-nitro-imidazole, we are going to react it with what? We are going to react it with this one. What is this? This is CH2CH2, this is ethylene. Now to this ethylene, what is that? Chlorine. So it is ethylene chlorohydrin. OH is hydrogen. So ethylene chlorohydrin. The hydrochloride will be released and you will get the final compound, and that final compound is nothing but the metranidazole. So what is happening? That NH at the position 1, we are supposed to have what? CH2CH2OH. So this is CH2CH2OH. This particular will get attached to the number 1, and this Cl and the proton which is present at the nitrogen will be removed as HCl, and you will get the final compound as metranidazole. So in short, please understand, metranidazole is the one of the important anti-protozoal drug. This metranidazole is having the 5-membered imidazole ring, and to this imidazole ring at the position 1 there is the hydroxyethyl group is attached, to second position methyl, and 5th position nitro group is attached. That is the very reason if you nomenclate the entire ring, that would be 1-hydroxyethyl, 2-methyl, 5-nitro-imidazole. So that would be your metranidazole ring. Now it is actually acting as a prodrug. That is, once it's entered into the system, what happens, that the electron transport chain causes this nitro converted into hydroxylamine. This hydroxylamine get bound to the covalently to the DNA of the protozoa, and that is a lethal effect. Even the reactive oxygens are released, so this will cause the destructive effect on the DNA or the proteins, membrane proteins. Understand? And during the synthesis, we have discussed, we are using 3 things as for the synthesis. One could be your glyoxal. What is glyoxal? 2-CHO, that CHO attached to CHO, then 2 amino and that is two NH3 molecules, and the third one will be the CH, that is the acetaldehyde. Now once these three things get attached to each other, you will get the first substitution, the cyclization, to get the 2-methyl-imidazole. Now once you get the 2-methyl-imidazole, now this 2-methyl-imidazole will undergo nitration reaction, that is the nitric acid with the sulfuric acid, and you will get what? Nitric acid with sulfuric acid will get the 5-nitro-2-methyl-imidazole ring that you will get. Now after that the substitution remains only at the position 1, so that what you are using CH2CH2 Cl and OH. So what is that? Ethylene chlorohydrin. When ethylene chlorohydrin react, the proton which is present at the nitrogen of the imidazole and Cl, they will be removed as HCl, and you will get the final compound as 1-hydroxy-ethyl 2-methyl 5-nitro-imidazole. That is nothing but the metranidazole. So this is about the metranidazole.